challenges in lima bean production. Specifically, his work focuses on quantitative genetic resistance to southern root knot nematode and white mold. Today, to discuss his work in plant pathology and genetics, please welcome Shem Msabila.
Howdy. I can see lunch is kicking in, so I want to ask you all to stand up, freshen up a little bit, and then we sit down and proceed.
Great call. Great call, Shem.
Perfect. First and foremost, I would like to thank you for inviting me down here, giving me the platform, listening at me, and also a great thank you to the organizing committee and the funding agency for giving me the travel award. All right, so my title today is Quantitative Resistance to Southern Root Nematode, Melodone incognito, identified in lima beans. So a little bit of background. Lima bean is an important commercial crop in Delaware, where it contributes roughly $10 million annually, and it also sustains regional agricultural livelihoods. It also makes Delaware the leading state when it comes to lima bean processed acreage, and also it has a profound nutritional profile offering fair amounts of protein, minerals, fibers, and vitamins. Unfortunately, it succumbs to several production challenges, and one of them includes the southern root knot nematode. So what are root knot nematodes? Root knot nematodes, these are parasitic roundworms which hijack resources required for plant growth and development. Root knot nematodes have a life cycle which involves the eggs, juvenile stages, and adult stages. A single adult female can produce up to 500 eggs in one setting. These eggs are protected by a gelatinous matrix which prevents desiccation and predation, enabling them to survive for several years in the soils waiting for favorable conditions. Their second stage juvenile is mobile. It locates plant roots and then gets inside to form permanent feeding sites. Its presence within the roots leads to formation of swellings or galls, or others call it knots. So now these knots prevents the uptake and transportation of water and resources from the soil upstream to other parts of the plant. So the key species affecting lima bean in Delaware is the southern root knot nematode, or scientifically known as Melodone incognito. So now, after knowing our problem, we came up with the objectives of this study, which were simple yet strategic. To evaluate resistance to southern root knot nematode in a recombinant hybrid line population, which was developed at the University of Delaware. And then our hypothesis was that resistance is polygenic, meaning it's controlled by several regions within the DNA. So these experiments were conducted in the greenhouse. I did these experiments in the spring and summer of last year. It involved the 8th generation of 2 parents which were crossed. One was susceptible and the other one was resistant. We had a total of 137 recombinant hybrid lines. So after growing them, initially in these containers, we introduced root knot nematodes and we let them associate for a number of weeks together so that we could associate them with resistance or susceptibility. So at the end of the day, on day 98, we did our final assessment of resistance by considering their extent of damage and the extent of reproduction. So the 2 traits that we assessed were the galling and egg mass count. Galling was done on a 0 to 10 scale adopted by Bridge and Page, 1980. And 0 means no galling, while 10 means severe galling, which eventually led to plant death, with the order of increasing number, distribution, and size of the galls. And then egg mass counts was done with the help of aeroglycin, which stained the eggs blue. And then we counted them using the mounted magnifying lens. So that is the data snapshot. On the top, we see these were the GAL ratings on 0 up to 9. We didn't have 10. And then below, we see these were the egg masses which were stained. And then this is a close-up view. You see 7 and 8, they're really devoured by the gals. And then these blue-like balls, these were the egg masses, which I had to count them individually. So now after having this data, our next step was data analysis. So the best linear unbiased predictors were developed to minimize blocking and replication effects, but also mixed models were adapted to adjust straight means. And we also did the composite index to unify the two traits to come up with one unified unit, which speaks more volume when it comes to resistance or susceptibility of these lines. But to avoid oversimplification and overprocessing of our data, we also subjected raw data to assessments. And what we can see first and foremost is that our two traits, the GAL scores and the GAL scores and the egg mass counts, they were highly correlated. And then the other thing is that in this graph, the x-axis is the recombinant inbreed lines. Again, they were 137, and the y-axis is the GAL rating from 0 up to 10. We see that we have a transgressive population. This is the susceptible parent. This is the resistant parent. This is the resistant parent, and then this is the resistant decile. This is the susceptible parent, and this is the susceptible decile. This is the population, mean, and median. And then this is for egg mass counts, which is pretty much plotted the same. We see that we had individuals which outperformed both parents on either ends. And we were able to cut down root-knot nematode GAL scores by 50% in the resistant decile compared to the overall population mean and median. And also, we were able to cut down egg productions by 70% in the resistant decile. While in the susceptible decile, we had up to 160% more eggs produced compared to the population mean and median. And we had about 80% more eggs produced. So having this data, we were on our way towards our QTL mapping. So requirements for QTL mapping is the genotype data, which we had it from UC Davis. So we sent them all 137 individual plant materials, and then they genotyped them and sent us back the results. They also provided us the genetic map, and I had the phenotype data from the greenhouse experiments, and I also created the configuration file to link everything together. After putting everything together, we identified a region of interest in chromosome 5, close to chromosome 6. So this is lambda chromosomes on the x-axis, 11 chromosomes, and on the y-axis is the logarithm of odds score. Now, this is on the basis of the composite score. If we are to assess each individual trait, let's say randomly, just the GAL rating, we still have our region of interest in chromosome 5. If we go for egg mass scores on random blocks, let's say block 3, we still identify our region of interest under chromosome 5. If we are to view it from a different angle, we see that still chromosome 5 is our region of interest. So now, this brings us to our discussion that first, our population had a strong genetic signal, and we also saw that resistance and susceptibility is hereditable. We also saw consistency between egg and GAL score data, and also our raw phenotypic data told us the same story with our statistically processed data. There has been a similar study which was done back in the days in 2008 by Roberts et al. And according to him, he had identified 3 genes which are responsible for resistance. And these genes were independent, controlled by different regions in the DNA. So now this brings me into future recommendations of reconfirming these QTLs and assessing trait correlation. And also do these experiments under real-world conditions in the field and be able to narrow down and identify the genes that are responsible for this kind of resistances according to the populations that we worked with. With that being said, I would like to acknowledge the funding agency, my lab cohort, my PhD committee, a UD research team, and of course, you all for listening. Thank you very much. Time for questions.
If you could in the future, would you be interested in, some sort of more high throughput method, or do you believe that it's just too difficult at this point to achieve such a goal without, I feel like I'm just begging the question.
Yeah, I mean, it was a tedious activity because some lines had up to 300 eggs, and I was working with a total of 1,244 plants because it's 137. Each one of them repeated a total of 9 times. So if there's a way to simplify the data collection aspect, it also increases the accuracy of the whole activity. I tried to look and see if we could be able to get some kind of programs to help us with that, but lima bean roots are a little bit tricky to work with. They're really, really fine, and it's just that the eggs distribution were very random, but I'm open to suggestions if any.
Have you tried any kind of 3D imaging at all?
Not per se. So, I mean, in the same lima bean program that I'm working with, we also have another PhD student who is working on some phenotypic data, especially on flowering. And with them, they're trying to use more of robotics to collect some of their field data. So I've pitched in the idea of maybe having some segments to help me with collecting the data on the roots, but still, I mean, the conversation is still moving in cycles. So maybe I can put more emphasis, much especially with all the exposure I got today and the brilliant ideas I also gained.
Do you believe, I know that lima bean is not very closely genetically related to a lot of other legume species. It can't interbreed whatsoever with Phaseolus vulgaris, for example. But do you believe that this could have potential ramifications for other leguminous species?
Yeah, right. Even this approach was borrowed from the common bean, Phaseolus vulgaris. And even before us being able to sequence the whole lima bean genome, we could rely and sometimes borrow some knowledge from common beans. So it is very relatable when it comes to other legumes. Now, what is special with lima bean is that it's mostly grown in Delaware. I would say it's among the top 5 most grown crops in Delaware compared to any other legumes. So if similar areas with the same problem, they could also borrow our results and approaches and be able to work on their challenges. So yeah, it can pretty much work both ways.
Thank you.
Right.
Hi, thank you for the presentation. I wanted to ask about the different parasites you were dealing with. So you talk about the species of nematode, but I'm sure that's not the only kind of nematode in the soil there. And I'm sure there's other pests besides nematode.
Right.
How is it that in your samples you were only seeing eggs from this species? How did you know?
Okay, that is a very good question. So I had to cut some of the information from materials and methods so that I can fit in the 10 minutes of this loss that I was allocated. But in setting up this experiment in the greenhouse, we did these experiments in these containers, and we used a media which was sand and premix mixed at a ratio of 1:1. So sand and premix, they were all sterilized. So there was nothing in it at all. And when I grew in the lima bean seeds, I introduced 500 nematodes in each container. So there was only nematodes which were introduced in these containers. So everything else was kind of controlled from the beginning and from the setup.
Okay.
Yeah, so there was no any mix-up. And even from the field diversity studies and assessments which we've done, we only have this one species up there. It's only the southern rudon nematode, Melodona incognita. We have not identified any other species, and we have done field data collection and sampling for 4 years in a row now. Right, so even like accidental introductions, they can happen because we only have that one species.
Yeah. All right. Thank you.
No problem.